Welcome back, students. In Part 1, we focused on autotrophs. In Part 2, we will focus on heterotrophs. As you work through the stations, think about the ideas that we introduced in Part 1 and really use them to compare and contrast autotrophs and heterotrophs. Now let's get started. Station G is about resource acquisition in eukaryotes. So eukaryotes have some very cool structures with which they can capture their food resources. We have some very cool models here that can show you these structures, but first let's look at a live example. So we have our sea anemone here. The sea anemone has these tentacles, and on them are stinging cells. Let's watch what happens when we put in a piece of squid. So now that it has engulfed the squid, it needs to extract those nutrients, and it will do that using its digestive system. So there are two types of digestive systems. We're going to talk about a two-way gut system first. So let's look at our sea anemone model over here. So in our sea anemone model, food enters through this hole, is digested in the digestive cavity, and waste exits out the same hole. Now, an important adaptation for nutrient absorption is increasing the surface area in the digestive system. How has the sea anemone accomplished this? So, if you look carefully, it has these accordion-like folds inside of its gut, and that increases the surface area. Let's talk about one-way gut systems now. So in a one-way gut system, food enters in through one end, is digested in the digestive cavity, and then exits out the other end. So it doesn't exit out the same hole. Now, thinking about that, what kind of gut do we have? So humans have a one-way gut, right? We eat food, it comes in through the mouth, it's digestive in the stomach, the digestive cavity, and then it exits out through your butt as waste. So in a one-way gut, there's a lot more potential for specialized structures to form. And what I mean by that is specialized digestive organs like the stomach and the small intestine. If we go ahead and look at our rabbit model over here, right? It has a stomach here, it has a small intestine, it has a large intestine. So keeping these specialized structures in mind, how does a rabbit increase its surface area for nutrient absorption? So in a rabbit, nutrient absorption primarily occurs in the small intestine. And you can see that that also has a lot of folds, a little bit similar to what the sea anemone had going on. Inside the small intestine, they will also have finger-like projections, which also increase the surface area. What is a heterotroph? That's the topic of this station. So heterotrophs don't make their own food. They have to feed on other organisms. So we're able to categorize heterotrophs by what they eat. So you may be familiar with the terms herbivore, meaning something that eats plants, or carnivore, meaning something that eats meat. But we can further categorize heterotrophs by how they feed. For instance, some animals, like earthworms, simply eat particles that are deposited on the surface. They are known as deposit feeders. There are also aquatic animals that feed on food particles that are suspended in the water. These are known as suspension feeders. Let's look at a few examples. We have two examples here. On the left, we have a feather duster worm, and on the right, we have some gooseneck barnacles. Their tiny tentacles curl around the suspended particles, capturing their food. Now, you don't have to be big or an animal to be a heterotroph. Here at this station, we also have 4 examples of single-celled heterotrophs for you to look at. Let's check them out. Station I focuses on how invertebrates feed. Invertebrates showcase a variety of different feeding structures. First, we're going to check out some chewing mouthparts. So this is a video a friend of mine shot. Thank you, nanonaturalists. So this caterpillar is using its mouthparts for chewing up the tough cellulose of leaves and stems. The mandibles allow the caterpillar to break down food so that it's easier for it to digest. In this next part, a caterpillar is excreting waste. So from this video, can you tell what kind of gut that caterpillars might possess? So let's go ahead and look at a predator here. So this is a scorpion. Ours has kind of holed itself up in its little cave here. So we've got a specimen. Instead, this is a real specimen, but it's dead. So scorpions also use their mandibles to crush up their food. However, how is it able to capture its prey? So if we look here, we can see that it has these really big claws. That's what it uses to grab its prey. And then it has a stinger, which it's able to inject a paralyzing venom into its prey in order to subdue it. Now, let's look at the mouth parts of a very interesting aquatic animal, the sea urchin. So sea urchins feed on algae that's growing on rocks. They also feed on kelp. But in order to scrape the algae off the rocks, they have a very interesting mouth part called the Aristotle's lantern. So if we take this urchin here, let's see if we tease it a little bit. See if it pushes the mouth out a little more. So that's the mouth part right here. It's kind of like a bunch of little teeth arranged into a little point. And this is what it looks like kind of inside the urchin. And so you can see here the little teeth that kind of form the kind of biting area, what it uses to scrape the rocks. And the inside we have the areas where the muscles will attach. So it kind of opens up in this motion and grabs the algae off the rocks. Lastly, we have another predator. We have a spider, a tarantula. And how the tarantula captures its prey is also by injecting a venom into its prey using its fangs, but it does something else. So digestion for tarantulas and spiders actually begins outside the body. They inject an enzyme, a digestive enzyme that dissolves the flesh of their prey into a more liquid form, and then they will direct this liquid into their mouths, which is behind the fangs. They don't drink with their fangs. They're not little vampires or anything like that, which also is a bit of a misconception because vampire bats also don't do that. Station J is about feeding specialization invertebrates. So we're going to talk about two different types of dentition today, homodont dentition and heterodont dentition. Let's start with homodont dentition. So homo means same and dont means teeth. So in animals with homodont dentition, in this case we have a gator skull right here, you have very similar looking teeth that are very simple. So you can see that here, they're just simple kind of little triangles. And animals with homodont dentition don't really chew or do a lot of kind of specialized feeding with their teeth. These teeth are meant to grab onto prey, and then usually they're swallowed whole. And that's very different from animals with heterodont dentition. So we've got quite a variety of skulls here for our heterodonts, or animals with heterodont dentition. First up, we have our dog skull here. So just a reminder that hetero means different, and dont, once again, means tooth. So they've got different kinds of teeth. So this is our dog skull here. It's an example of our carnivore. You have the sharp pointy teeth here that are meant to grab their prey, right? But the back teeth here, they kind of function you see kind of like a scissoring motion that is very good for tearing and breaking apart meat into small sizes so that they can swallow that. But you can see these teeth would not be so good at breaking down some tougher materials like vegetation. And for that, we are going to go to our herbivore skull here. This is a horse skull. And for the horse skull, you could see, first of all, the incisors here. Very good for clipping grass. And then we have the molars. Look how flat they are. They're very good for grinding vegetation. And then last of all, we have our macaque skull. So the macaque skull is interesting, right? So you've got the big pointy teeth like you would in the dog skull. But you also have the flattened molars, and that's because macaques are omnivores. So they're able to eat both meat and vegetation. And if you look at your teeth, if you look at your own teeth, you would find the same thing. Station K is about the fungus among us. In particular, today we're going to be talking about saprotrophs and parasites. Let's start with saprotrophs. Saprotrophs are organisms that feed on other decaying organisms. We'll be using mushrooms as an example. So most of us are familiar with mushrooms. These are oyster mushrooms, a type of mushroom that we eat. And then there's also, plenty of mushrooms that you can't eat because they're poisonous. Mushrooms, though, are not actually the digestive part of the fungi. Mushrooms are the fruiting body. This is how fungi reproduce. Most of the action for digestion takes place underground, where the hyphae are at. So if you look here, there's like a few little tendrils that are the hyphae of the mushroom, and that's what they'll use to go and break down decaying matter. So this is important, right? Because fungi feed on other organisms. We look at fungi and they kind of resemble plants, but we have to remember that they are not plants, they are not producing their own food, they are heterotrophs. So another example of fungi here that we have is brown wood rot. In brown wood rot, you can see this was collected in the Sierras, and it's still actually digesting the wood here. You can see how it's breaking it down into fine, fine particles. So all the lignin has been easily digested, but the cellulose, which is harder to digest, has been left behind, and that is what we have here. So let's transition now from fungi to parasites. Not that they're mutually exclusive. There are some parasitic fungi. If you've ever had athlete's foot or ringworm, you've had parasitic fungi. Now, the parasites we're going to look at now are not live specimens, but I'm going to make up for it in, shall we say, grossness, or perhaps you'll find them very exciting like I do. These are botfly larvae. So botflies are a type of fly, and their maggots will burrow into the flesh of things and basically live there until they're big enough. Then they drop to the ground, pupate, and become another adult botfly. So this is the botfly maggot of a type of botfly that lives in rabbits. And this jar here, is a whole lot of botflies that occurs in horses. These are attached to some stomach lining. And although these can be irritating and painful for the organisms that they're living in, it does not kill the organism. They are feeding off of a living organism, and that is what parasites do. Station L is about respiratory systems and gas exchange. So the idea of how to capture oxygen and then how to distribute that oxygen throughout the body. Now, a theme running through Lab 2 is the idea of surface area to volume ratio. And that is especially important for gas exchange. And I'm going to do a little demonstration to show you why. Now I have 2 balls of clay here about the same size. I'm just going to go ahead and flatten one right here. And by doing that, I have increased its surface area. Now it's pretty easy to pick up oxygen through diffusion with a lot of surface area. So comparably, you could see that it's very thin, right? So if you're able to pick up oxygen from all of your flat surface area, and then it's very easy to also distribute that into the center, because you can absorb oxygen from here and then just distribute it right through your thin center here. Now, if you're more of this shape and you are picking up oxygen, first of all, you have less surface area than this model here. And then you also have the problem of having to move that oxygen into the center of your mass. So there are 3 main ways that an organism can capture oxygen. And these are diffused through the skin, captured in the water through gills, or captured from the air through lungs. And here we have an example of each that we are going to walk through. So first up, we have a feather duster worm. And in these models, the respiratory system is highlighted in red. So in the feather duster worm, which you have seen, these fronds were used for capturing food particles, as it was a suspension feeder. But these fronds actually do double duty. They are also what it uses to breathe, to diffuse oxygen through, because they're very thin, right? So that's a way to increase your surface area, is to create really thin kind of projections, fronds or folds, to really just pick up more oxygen. Now over here we have a different type of way to capture oxygen. We have the gills. So in crabs have gills at the base of each leg. Why do you think that it's advantageous to have your gills at the base of each leg if you are a prey animal? So if you're a crab and your gills are attached to your legs, and you lose a leg, you would still have 9 other gills and 9 other legs, and you'll be able to breathe just fine. So our last example here is the rabbit. And the rabbit is like us. It has lungs. It inhales oxygen, it exhales CO2. When the oxygen is taken up into the lungs, it is distributed throughout the body by the circulatory system. Why do you think it's important for a rabbit to have a circulatory system? And why doesn't something like a single-celled heterotroph, like a blepharisma, have a circulatory system? So if you're small, you could easily diffuse oxygen to all of your cells. But if you're larger, it's harder to distribute that oxygen throughout your body. And that is what a circulatory system is for.
That's it for this video. As always, if you have trouble with any of these concepts, please contact your TA. Thank you for watching.